Hi, it's Tom with Biology Magnets, and this video is over lesson 10A, G protein signaling. And this module has magnets that show a lot of aspects of G protein signaling pathways. And I've got kind of all of them set up here for the first lesson A. And what we have here is, the first part I'm going to show you is how epinephrine causes glucose to be released from cells. And it utilizes receptors in G proteins. And here is our epinephrine molecule. The epinephrine molecule will bind to a G protein coupled receptor, which is this receptor. This is the cell membrane, which I've drawn with a marker. And here's the G protein coupled receptor. And it receives the epinephrine, and that fits in there like that. And this protein is actually coupled to a G protein. Now this whole thing here is the G protein. The G protein has two subunits, the alpha subunit and the beta gamma subunit. This is a GDP molecule, guanosine diphosphate. There are the two phosphates. There's the guanosine. And the guanosine diphosphate molecule will stick to the alpha subunit. And this is, the alpha subunit is not activated yet. But when the epinephrine bonds to the G protein coupled receptor, it causes a conformational change where the alpha subunit of the G protein releases the guanosine diphosphate, and a guanosine triphosphate will attach in its place. And so that's why it's called a G protein, is it's activated by this GTP molecule. And when that happens, very often the alpha subunit and the beta gamma subunits will break away from one another. And they'll travel through the membrane. And that's what happens in this case with epinephrine. Now, the alpha subunit and the beta gamma subunit, they have these lipid tails that stick in the phospholipid bilayer of the cell and keep it associated with the membrane. So they don't, because of these tails, will not come away from the membrane. They stay in the membrane. And all of these proteins right here that I have in the membrane have these lipid parts that stay associated with the membrane. So they stay attached to the membrane. So what happens next is the alpha subunit comes over here, and it attaches to this protein, which is called adenylyl cyclase. And that switches on the adenylyl cyclase molecule. And what that adenylyl cyclase will do is it'll take ATP molecules. Now I have a bunch of ATP molecules at the bottom here, and you can use them in explaining this. But the ATP attaches to the adenylyl cyclase, right here at the active site. And the adenylyl cyclase will tear apart the ATP, will take off the phosphates on the ATP, and will form a new molecule called AMP, adenosine monophosphate. And it's actually called cyclic AMP because there's a bond that forms between the phosphate and the sugar. You could draw it on here if you were motivated to do that. An extra bond forms. And so it makes it kind of cyclic. And this cyclic AMP molecule is what we often call a second messenger. We have the reception of the signal, and then this stuff that goes on the inside that leads to these second messengers is called transduction of the signal. And what these second messengers like cyclic AMP will do is they'll cause further effects inside the cell. So in this case, the cyclic AMP molecule comes over here and it binds to protein kinase A, which is a large protein that actually has 4 subunits. And we'll need several of these ATPs to be broken down into cyclic AMPs. I'll draw my little cyclic portion, why not? And we'll just use two of them here. They both attach to the protein kinase A. I'll move these phosphates out of the way. Now this protein kinase A, when those cyclic AMPs attach, actually breaks up. And it's now in 4 pieces. And what occurs next is called a phosphorylation cascade. ATP molecules will be used to phosphorylate the catalytic portions of the protein kinase A. So protein kinase A has regulatory portions that the cyclic AMP attached to, and it has catalytic portions that are phosphorylated by ATP and become active. And these catalytic portions will phosphorylate other proteins. And the way they phosphorylate them, again, is using ATP. And I actually have a site on the protein kinase A here where the ATP can attach, if you'd like to do that. And the ATP, if you'd like to show that, the ATP attaches and then will pass on a phosphate to the next protein in the phosphorylation cascade, which happens to be phosphorylase kinase. And then that will in turn phosphorylate another molecule. So that's why we call this a phosphorylation cascade. Another ATP comes in and attaches, and its phosphate is passed on to this next protein, which is called glycogen phosphorylase. And so we're passing phosphates on from basically from one molecule to another. Or a phosphorylate, we're not passing phosphates, but we're phosphorylating one molecule after another. Now each step of the phosphorylation cascade can increase the activity in the cell. For instance, this protein kinase A doesn't just phosphorylate one phosphorylase kinase, it can phosphorylate a whole bunch of them. And in turn, the phosphorylase kinase can phosphorylate a bunch of these glycogen phosphorylase molecules. So every step of the phosphorylation cascade has an increasing effect on the numbers of molecules that are active inside the cell. And you'll see that go way up in an exponential growth curve inside the cell. Now, I don't have enough magnets to show all that, but you get the idea. This final protein, glycogen phosphorylase, will go to glycogen, which is a molecule stored in your liver cells. This whole cell could be a liver cell. I probably should have said that first. And it's storing glycogen. And so this glycogen phosphorylase can one by one break down that glycogen into glucose. Now in actuality, it's also phosphorylating the glucose each time. So you could show an ATP attaching to the glycogen phosphorylase and putting a phosphate onto the glucose and the glucose breaking away. That phosphate will later be removed, and these glucose molecules can exit the cell. And in that way, epinephrine causes the release of glucose from the cell. It's, as you can see, there's a reception portion, there's a transduction portion, and then there's a response portion at the end, where the glycogen phosphorylase molecule is breaking down the glycogen into glucose. The response is the release of glucose. And that all happens pretty fast. Just within a few seconds, you can get glucose to be released. Now, it may be easier for you not to use all these ATPs. I like, when I'm teaching it, I actually just kind of use the phosphates, and I'll say this one causes phosphorylation of this one, and this one causes phosphorylation of this one. And so you're not having to muddle up your board with too many magnets. It could get a little confusing, but depends on how you want to teach it. Now, a couple other things I want to show. The beta gamma subunit that was released can go on to also cause other things to happen in a cell. Now, some cells use the beta gamma subunit to activate a channel protein called a GERK channel, which the GERK channel actually lets in potassium ions. The beta gamma subunit can attach to the GERK channel. The GERK channel protein can open up, this shouldn't be here, and let potassium into the cell. That's one way a cell has of getting in potassium, is having a ligand. It's not necessarily epinephrine in the case of the GERK channel, but the ligand binds to the receptor, the beta gamma subunit is released, and it causes this GERK channel protein to open and let potassium in. So the beta gamma subunit can also activate proteins just like the alpha subunit can. Now, there's another instance where the alpha subunit can attach to a protein called phospholipase C and activate that protein. And this alpha subunit phospholipase C complex can break down a complex that's in the membrane that consists of two molecules, DAG and IP3. This complex is broken down by this PLC molecule that fits in like that and breaks it apart. And this IP3 can act to open channels attaching to this calcium channel. This is the endoplasmic reticulum, and the calcium channel will open up and let calcium out of the endoplasmic reticulum. And the calcium can act as a second messenger and cause things to happen in the cell that way. Many biology textbooks talk about the DAG IP3 pathway. So your textbook may teach that. So you can use these magnets to teach that as well. Now there's something else I wanted to show. This module has ways of showing regulation of the process. So one form of regulation is a protein that sits in the membrane called the RGS protein, the regulator of G protein signaling. So the RGS protein can move about in the membrane and can attach to the alpha subunit. This causes the GTP to lose its phosphate, or we say hydrolysis of the GTP. The GTP can lose the phosphate, and now that turns off its activity. It will no longer bond to whatever it's bonding to, because when it loses the GTP and goes to GDP, it changes its configuration and stops binding and stops activating the proteins it does. So that's the RGS protein. Another way we can stop the activity of this signaling pathway is using this protein that is called a G protein coupled receptor kinase. Now, if you remember, like this protein kinase A over here, a kinase phosphorylates other proteins. So this GPCRK, this kinase, will come up, attach to the G protein coupled receptor, and phosphorylate it. Now, it actually phosphorylates it by having ATP drop off phosphates, but I just like to show it like this, dropping off phosphates there. You don't have to show it that way, but you can. And now this receptor has been phosphorylated. And another protein called arrestin can come in and bind and basically get stuck where these phosphates are. And now this G protein coupled receptor is turned off. And it cannot activate any more G proteins. If a G protein comes up to it that has a GDP in it, it cannot stick to the coupled receptor and be activated because it's been turned off. That's a form of regulation. The arrestin protein sticks where those phosphates are and can shut the whole thing down. So there's a lot in there. Different textbooks talk about different aspects, but you can show a lot of the different ways with these magnets, and that's a good way of teaching cell signaling. If you want more information or more instruction about this, go to biologymagnets.com.